Hello, my name is Bobby Gaspar, and I'm the CEO and co-founder of Orchard Therapeutics. I'm delighted to have this opportunity to speak with you about Orchard and our vision to make a lasting difference in severe genetic diseases through gene therapy. While Orchard was founded in 2015, our roots and commitment to advancing the science of gene therapy go back decades. My own journey in the field of gene therapy began in 1992. I had just started as a junior doctor at Great Ormond Street Hospital for Children in London, and this is where I first worked with children suffering from devastating rare diseases. The very next year, I was involved in one of the first clinical studies of gene therapy. When I first heard about taking a child's own blood stem cells, introducing a working copy of the missing or faulty gene, and then returning the gene-corrected cells back to the child, I thought this was science fiction medicine, and that my professor at the time was playing a joke on me as a new junior doctor. Little did I know at the time, bringing this seemingly unbelievable medical advancement from the academic to commercial setting would become my life's work and the focus of Orchard. The science has come a long way since then. Today, we're at the cusp of an exciting new phase of advancement. We have the potential to make gene therapy commercially available, offering a one-time, potentially curative therapy for devastating genetic conditions. At Orchard, we believe that hematopoietic stem cell, or HSC gene therapy, has the potential to transform the lives of patients with genetic diseases. To date, we've treated more than 160 patients across 5 diseases using the HSC gene therapy approach, and we possess more than 10 years of follow-up data in the earliest treated of those patients. Our understanding of the biology of the disease, the mechanism by which HSC gene therapy exerts its effect, and the clinical validation in rare diseases gives us confidence to take this approach into larger indications. We're excited to share more about the HSC gene therapy approach that Orchard employs, the groundbreaking science that underlies it, and how it informs our sense of urgency to create lasting change for patients and families facing devastating genetic conditions. Hematopoietic stem cell, or HSC gene therapy, leverages the power of a patient's own genetically modified HSCs to correct the underlying cause of a genetic disease in a single administration. HSCs are responsible for lifelong sustained production of all blood cells, and they have 3 characteristics that make them ideal for targeted gene therapy. 1, they're capable of self-renewal. 2, they can give rise to multiple cell types in the bloodstream. And 3, they're able to naturally migrate into multiple different organ systems, including the brain and GI tract, as tissue macrophages. In the HSC gene therapy approach Orchard uses, we insert a working copy of the gene into the genome of the patient's own HSCs, so that once engrafted, every time the cell replicates, that working copy of the gene is passed on. This offers stable engraftment of gene-corrected HSCs, which is expected to be permanent. Importantly, a subpopulation of HSCs can develop into cells that have a natural ability to cross the blood-brain barrier and distribute throughout the brain. Once in the brain, these cells further differentiate into specialist cells called microglial-like cells. Because these cells have been genetically modified, this gives us a route to deliver therapeutic genes to the CNS, and that allows us to correct defective cells in the brain. We are on the cusp of an exciting new era due to the transformative potential of HSC gene therapy to offer durable delivery of gene-corrected cells to a range of tissues and organs, including the central nervous system and GI tract. Now that we've addressed the importance of HSCs, let's take a closer look at the process. Before I share more about the HSC gene therapy approach that Orchard employs, I'd like to speak broadly about gene therapy. At the most basic level, gene therapy is the delivery of a therapeutic gene to correct the underlying cause of a genetic disease. It's important to note that there is no best approach to gene therapy. We like to think of gene therapy as multiple different tools, and we have to think of what is the appropriate tool to treat a specific disease. At Orchard, our goal is to develop HSC gene therapies that provide durable clinical benefit after a single administration. This is a highly differentiated approach that provides the potential for permanent disease correction of specific conditions. Now let's talk specifically about the steps involved in the HSC gene therapy approach that Orchard employs. First, a patient's own HSCs are collected, either from bone marrow or mobilized peripheral blood, and the blood stem cells are then selected and purified. Next, the cells are transduced outside the body with a viral vector being used to insert a working copy of the gene into the patient's own cells. Then the patient is conditioned to receiving the treatment. And finally, the gene-corrected cells are infused into the patient. Because the gene-corrected cells self-renew, a single administration of gene therapy has the potential to create lasting change for patients. Just think about that, a one-time administration that has the potential to result in a lifelong therapeutic effect. Just take a moment to imagine what this could mean. While we've initially focused on rare genetic diseases, we see the potential to apply this gene therapy model to other genetic conditions with larger patient populations. With this in mind, we aim to make a lasting difference for patients and families facing a variety of devastating diseases. Let's take a look at one of the larger disease areas we're working on now. Genome sequencing and genetic association studies have enabled great strides in our understanding of the genetic cause of a disease. This, coupled with Orchard's foundational understanding and proof of concept work in small, rare diseases, has led us to undertake investigational research programs in larger indications. As an example, let's look at Crohn's disease. In the case of Crohn's, the ultra-rare model that we first understood was chronic granulomatous disease, or CGD. This is a rare, life-threatening, inherited disease of the immune system in which white blood cells are unable to kill bacteria and fungi, leading to repeated chronic infections and severe colitis. In some cases, the colitis in CGD looks just like colitis in Crohn's disease. So we began to explore the implications for larger conditions such as Crohn's. For Crohn's, mutations in the NOD2 gene are associated with a severe form of the disease. In people with the NOD2 deficiency, the macrophage cells in the gut wall can't appropriately sense and respond to bacterial infection, ultimately leading to colitis, a hallmark of Crohn's. This is where we see an opportunity to potentially correct this subset of colitis with HSC gene therapy. We believe our gene-modified HSCs can become gene-modified macrophages, which contain functional NOD2 and replace the defective macrophages. The functional macrophages can now sense bacterial invasion and respond appropriately, preventing colitis. Based on this, Orchard is investigating the HSC gene therapy platform to potentially deliver the full coding sequence of a functional NOD2 gene and correct the underlying genetic cause of the NOD2 form of Crohn's. Crohn's is just one example of how Orchard aims to take the HSC gene therapy applications in rare conditions and apply them to larger conditions to further help patients living with genetic disease. An important piece of bringing gene therapies to patients is scaling manufacturing from investigational studies run by academic laboratories to commercial operations that meet the expanding number of potential uses with larger numbers of patients. To achieve this, Orchard is pursuing innovations in vector production and stem cell transduction. Let's start with vector production. The current manufacturing process meets quality standards, but it is a labor-intensive process. Orchard is exploring innovations to automate the process and identify opportunities for greater efficiency. For scalability, an important goal is to generate a clone or cell line in which all of those components of lentiviral vector are stably integrated. Once produced, the cell line can be cryopreserved as a master cell bank, so that for every batch of lentiviral vector at a basic level, we can thaw a vial and expand that clonal packaging cell line. Utilization of a stable cell line provides an opportunity to generate lentiviral vector of consistently high titer and eliminates the need to purchase plasmids prior to production of each viral vector batch, providing more efficient production processes and shorter lead times. Next, let's turn our attention to drug product manufacturing and the innovations that Orchard is striving to achieve in the area of transduction efficiency and drug product manufacture. Currently, the transduction of hematopoietic stem cells requires large numbers of viral particles to effectively transduce those HSCs. Achieving a more efficient transduction of HSCs could reduce the amount of vector needed to create the same therapeutic effect. In our laboratories, we've developed proprietary transduction enhancement cocktails that can help us achieve a 75 to 90% reduction in the vector we need to create the therapeutic effect versus the standard protocol. Ultimately, our aim is also to move to fully closed, automated drug product manufacture. These are exciting times at Orchard. We're inspired by the power of HSC gene therapy to offer a potentially curative therapy for a variety of devastating genetic diseases. We know that taken together, all the innovative advancements we're currently making, along with those still on the horizon, will allow us to make a lasting difference.